going to be doing the lab called mystery powder. This is kind of what your setup will look like. This is your well plate where you will be testing your solutions. Your directions tell you to load up your well plate in a labeled means. If you look closely at your well plate, it's actually numbered for you. 1, 2, 3, 4, and 5. That's etched in there. And you have 3 rows, of course. Now, the 3 rows correspond to your testing solutions. So later on you're going to put testing solution 3 in the 3rd row. You'll have your testing solution 2, which is vinegar, in your 2nd row. And you'll have your testing solution 1 all the way across your 1st row. Okay? So you're going to start out by loading up your well plate. To get your spatulas out, you may want to use a toothpick to get those out. And then you'll take your spatula and kind of scrape it across the edge. And you'll put sample A all down in here. And that's about as much as you need there. Okay, and then you will actually do that again all the way down. Okay, so A is going to go here. It'll go in row 1, 2, and 3. Okay, don't need a whole lot. Okay, so then you'll go sample B. Okay, we're going to put that in the 2nd column, and you'll do that for rows 1, 2, and 3. Now, you're getting kind of close to the other rows, so at this point you may want to be a little more careful in tapping these powders in, because you don't want to contaminate one well to the other. Okay, alright, so we'll go ahead and load up our C. You can see that I've kind of made a mess there, so that's another good reason to clean off your spatula before you put your solution in. Okay. Alright, so D and E right here. Okay. And finally E. Okay, after you get your samples loaded up, your data sheet and your instructions ask you what's your initial observations of these powders. So before you add any testing solution to them, you're going to look at the powders and you're going to make your observation about how they appear to you. Do some of them look like they have a larger particle size? Are some of them puffy, grainier, finer? So look at that particle size and you will write down, how does A look? You know, and so this is A. All of these are A. How does A look compared to B? And how does B look compared to C? So you will just kind of write in words. That's your qualitative observation. How do they look to you? Okay? Alright, so the next thing you're going to do is you're going to test all your solutions with water. Your directions will tell you to give it a little stir with your wooden splint, which is a toothpick. Now, if you do this, you're going to need a different toothpick each time you stir, so you don't contaminate your different wells. However, our solutions are designed in these bottles so that when you drop them out, you can kind of mix your solution up just by giving it a little squirt. Okay, so this alleviates you having to stir and use so many toothpicks. So give it just a tiny squirt. I probably added just a bit too much to that first well, but give it a little squirt as a means of stirring it. Okay. Once you add solution 1 all the way across here, you want to stop and look what you observe each time you add. Okay, and then you'll write what happened with A when I mixed it with water. Okay, so stop and write down your observations every time. If you just add water to all of them and then you go back and try to write down what happened, there maybe have something happen that you missed. So add your water to each one, look at it, and then write down what you observe after each test. Okay, so a few other things you have, and guys, so what you'll do is you'll do solution 2, you'll add it, a few drops in, this one, and then you'll do the same thing all the way across. And of course you'll do that with solution 3, which is iodine, all the way across here. Okay, you'll put, for instance, solution 3, this is iodine, here's sample E. So whatever you see happens here, you'll write down what happened in this column on your data sheet. Okay, so testing solution 3, sample E, the results would go here. Okay? Now, another way for you to hypothesize about what type of powders you may be dealing with is to carry out some additional tests. Your kit comes with red litmus paper, and you probably already know that red litmus paper stays red in the presence of an acid. Okay, so this is lemon juice. It has a low pH. It's going to remain the same color. This is ammonia. Ammonia has a high pH. I put a little bit in the cap. And so it changed colors. So once you test some of these solutions out, depending on what type of powder you think it is, you may have some information about the pH of that powder. And you can test the pH by also using blue litmus paper. Now, in the presence of a base, the blue will remain blue, but if it is mixed with an acid, it will change colors. Okay, so it goes to red. Okay, and you also have a third way for measuring pH. You have a strip of pH paper that you can put here, and then you can match that up to the color chart here. This would have a pH of somewhere between 10 and 11. The lemon juice pH, it's a lower pH, so somewhere between probably 2 and 3. Okay, so you can also use your litmus paper and your pH paper in your testing solutions to give you a little more information about what you're dealing with, to come up with what type of powder you may have. Once you finish all of your tests, you will write down which powder you think you had for A, what did you think you had for B. Okay? Enjoy.